really as well known as most of the other types of microbes, but they are significant in healthcare. And so I give you these statistics just to try and show that. You see that candidiasis is a group of yeasts that are very important in healthcare associated infections, because we have a lot of candida on our skin. So candida and some other yeasts are an important part of our microbiome naturally. They usually don't cause us a problem in healthy people. They're a good part of our microbiome. But when people are sick, they're more at risk of opportunistic infections, and so things that are in our bodies normally can become a pathogen. And so we'll talk more about how that happens in a minute. Another area with fungi is hypersensitivity and allergic reactions to especially to molds like aspergillus and their spores that we breathe in all the time. So that's a big area where fungi are medically important. And then also just infection with molds. They mention here just statistics on aspergillus, but there's a lot of other molds that cause infection and cause hospitalization and serious illness. So when we're talking about the yeast just now, and in the hospital setting, part of the problem with that is the use of catheters, and especially central lines. If you don't know, central lines are a type of catheter that are placed in a larger vein, usually in the chest or the upper arm, so more near the heart. It gives the doctors a way to give you multiple IV fluids when a patient is really sick. So that's a good thing, but because they're going through your skin and you've got a lot of yeast on your skin, they can be contaminated with yeast. And then that yeast gets into the bloodstream and can cause septicemia. So that's a big issue. And normally, patients that need central lines are immunocompromised people. So that adds another big risk factor there for yeast septicemia in hospitalized patients. Then look at these other categories. If you think about the way medicine has evolved in our lifetime, especially in my lifetime, we have so many more treatments now to help patients that need an organ transplant, or that have cancer, or an autoimmune disease. And those treatments involve suppressing your immune system. They're helpful treatments for these conditions. If you get an organ transplant, you certainly don't want your body to reject that. And the only way to keep it at this time from rejecting it is to suppress the immune system to some extent. And if you've got an autoimmune disease, you need a treatment that will cause your immune system not to attack your own tissue. So but that also leaves you more open to infection. Fungi, for the most part, they're kind of happy out in our natural environment. They don't need us for the most part. I'll give you a couple of exceptions as we go along, but they don't need us as a host. But when you have people that are immunosuppressed, and we are in the same habitat as the fungi, which mainly live in the soil, now except for the yeast, which are, they do live on our bodies, but the other molds don't, and they would be happy staying away from us, but because people are immunosuppressed, we're breathing in spores, and we're much more likely to develop infection when our immune system can't take care of those spores. Untreated HIV infection is another example. That's not so much an issue now since we have treatments for HIV. When HIV was the epidemic first began back in the 80s, that was a huge thing because those patients were incredibly vulnerable to infection too. So here's some other reasons why we have more fungal infections today than we used to. Just our environment is changing some. We seem to have more extreme weather and natural disasters. So why would that be an issue with fungus? Well, again, a lot of them live in the soil or the dust or the sand or some outside source like that. And when you have extreme weather stirring all that up, it puts more spores in the air. So we're more likely to breathe more of them in. We travel more, so we're exposed to that. Some fungi are endemic to a certain area of the world that we may never have come in contact with, but because people travel so much now, they're coming into contact with more things they're not used to being around. And then as we have more construction, again, stirring up the soil, aren't we? So that's a cause of the spores coming into contact with those. And just like farmers now use it, or I think they're kind of trying to get away from it, but they tend to use antibiotics on animals, farm animals. They also use antifungal agents on their plants. Fungi, while they really don't, we're not one of their primary targets for being a host, there is a lot of infection with plants. They do infect plants a lot. So the use of these azole antifungal agents in agriculture, what do you think that might do? The way we've talked about, when you use antibiotics a lot, what happens?
You become resistant.
That's right. And the same thing is going to happen with these antifungal agents. The more they're used, the more it gives the opportunity, the fungi, to try and find a way to get around them and to not be affected by them. So you've got now an issue with fungal resistance. All right, let's talk a little bit about what fungi are then. That's why we're going to be concerned about them from a medical standpoint. So they are eukaryotic organisms. So we've been talking a lot so far about prokaryotes, haven't we? But fungi are eukaryotic. So they have the true nucleus. They divide by mitosis or meiosis. They, as we'll see in a minute, they have two forms of reproduction. Most fungi can produce asexually by mitosis, or they have a sexual reproduction form that they can use as well. They have rigid cell walls, so they're a eukaryote with cell walls, and those cell walls have chitin and glucan as major components. That makes the cell wall quite strong, those two components. Those are polysaccharides. The components are fairly unique to fungi. I mean, those particular formulations of chitin and glucan. And also notice that the cell membranes have ergosterol instead of cholesterol, which we have in our cell membranes, don't we? To give them a little bit of flexibility and strength. So that chitin and glucan in the cell wall and ergosterol in the cell membrane, all three of those can be targets for antifungal agents. So that's something to keep in mind. We have developed, I can think of more than one class of antifungal agents that target the ergosterol And there are also antifungal agents developed that target the chitin and the glucan in the cell wall. All right, well, fungi, they get their nutrition. They don't have chlorophyll. They're not photosynthetic, so they're heterotrophic, and they do need organic carbon compounds for nutrition. They have the ability to break those carbon compounds down. They secrete a lot of enzymes, so they're major decomposers. They will degrade wood. They can degrade the cellulose and the lignin in wood. And then in that process, they release CO2 and nitrogen back into the soil, or release CO2 into the air and nitrogen into the soil. So they play a huge part in our environment by recycling organic compounds. So then the plants can use those, the CO2 and nitrogen, to continue that cycle. Bacteria do that as well, but fungi play a really big role. Another good term to remember is saprophyte or saprophytic. That means that the nutrients that they find in the soil through the wood and other things they decompose, they can use that for their nutrition. So they absorb those things, and we call that then, if the organisms that can do that are saprophytes. Okay, so when we think about fungi, of course, mycology just means the study of fungi. We talk about yeasts and molds, and those terms mean a yeast is a single cell, always. It's unicellular. Molds are multicellular, and sometimes we refer to them as filamentous. They have hyphae, which I'll go over in just a minute. You can see there, so this picture on the left is our unicellular yeast. This is a potato, actually. I don't know if you can tell that, but there's a mold growing there in the center. And you can see it's got a little bit of fluffiness to it. And I know you guys have all seen molds growing on various food products before, so you know how they look. So those are two types of fungi. Now, those are not used for classification. Classification in mycology is very complicated. I'm not going to delve deeply into classification. Hope that's okay with Dr. Daft. But because it's kind of in a state of flux right now. They, in the past, have attempted to classify molds and fungi, I shouldn't say, they all fungi, based on the sexual reproduction form. And that's been very difficult because we don't see the sexual reproduction form sometimes in the laboratory. There are some fungi, we have no idea what their sexual reproduction form looks like. So it's very difficult, and they are, I think, now using a lot more molecular techniques to try and classify, but we're still kind of in the process of working on that. But these are shapes that fungi have, the single cells and the filamentous fungi. So we'll talk about yeast and molds. Mushrooms are not, I don't consider them, they're not a type of fungi. That mushroom is just a structure that some fungi produce. And it is, that is a sexual reproduction form. If I can get I think that's a poisonous mushroom they're showing there. So not all fungi produce mushrooms, okay? It's just certain types. And normally they're not the ones that are very clinically significant. Okay, and I've already mentioned spores, and I think you know what those mean. The spores, fungal spores, are different than endospores that Dr. Daft has talked about in class. But they are not as hardy and resistant to drying and heat and things as endospores, but they are still pretty resistant and more difficult to kill than other types of cells. And I just mentioned that we don't see the sexual reproduction structures often in the laboratory, and it's because the fungi just use their asexual process much more often. It is very successful in distributing spores in their habitats out in nature. And so that's what they use because they're successful with it. And that's what we tend to see in the lab, and that is often how we identify them. I'm going to start with a little bit about yeast, and then we'll talk more about molds. So they reproduce, really, their reproduction is asexual. And it's just a form of mitosis, and we call it budding because you see there in this top picture, you've got your nucleus, and you start to have a little softening of the cell wall in an area of the yeast cell. And then mitosis begins, and you see the nucleus divides, and microtubules form, and that one of the nuclei then is taken into that new part of where the budding is occurring. And then that breaks off that bud, and you have a separate cell. Here's a microscopic picture of how it would look under the microscope, that budding that occurs. And so anytime we can see the cell, sometimes if you just see a single yeast cell, it might be a little bit hard to identify it as a yeast. But when you see the budding going on, you know you have a yeast cell because they're really, I think, the only things that can do that. So when we identify yeast in the lab, we can look at how they're growing on the plate. That's what we mean by colonial morphology, on an agar plate. Hopefully you guys, have you seen some pictures of, you're looking at bacteria, how they look on plates in your lab time, I guess. And yeast look fairly similar to bacteria when they grow on agars. And I'll show you a couple of pictures in just a minute. And then we look at them under the microscope to see if they are budding and if they're, shaped like a yeast. Then we can do biochemical testing on yeast, just the same way as bacteria. And do you know what I mean by biochemical testing? Does anybody have an idea? Yeast will ferment sugars just like bacteria. So that is some yeast ferment some sugars, other yeast ferment others. So that's one way we can tell them apart. There are enzymes that they produce. Some produce some enzymes, some others. So we can do a biochemical panel on them just the same way as we do with bacteria to see if we can identify them. And then there's also DNA sequencing now that we can use to learn which yeast we're working with. So this is probably the most commonly isolated yeast in the lab. We have a lot of it on our bodies. It's in our microbiome. And this is kind of how it looks on a blood agar plate. And you see it's a white colony. It's got those little, we call them feet sometimes. They're little extensions that come out from the colony. There are a few other yeasts that can do that as well, but Candida albicans tends to do that on blood agar. This is how it might look if you put it on a special media we call cornmeal agar. Do you see these structures here? Well, first of all, you can see there where you've got a yeast cell that's budding. So all yeast will look similar to that. But we have these long structures here. Sometimes the yeast cells can elongate and it almost looks like hyphae forming, so we call it pseudohyphae. This structure at the end of the pseudohyphae is called a terminal chlamydospore, which is kind of a misnomer because it's not actually a spore. It's just a structure that forms, that Candida albicans specifically forms on this type of agar. So it's very, we can almost say for certain it's Candida albicans when it forms that big structure there. A lot of yeasts also, this is more pseudohyphae, and then they will also form these smaller yeast cells that we call blastoconidia. I'm just going to mention one other yeast, and we'll go on to molds. This is kind of one that's really clinically significant for immunosuppressed people. Normally, healthy people don't have an issue with this. This is not part of our natural microbiome. We don't carry this organism, as far as I know anyway. I don't think so. It's anywhere really on our body. We don't have some sort of symbiotic relationship with this yeast. It is found out in nature. It is around, it grows in the soil around like pigeon droppings and bat droppings. Tends to concentrate. But if you're immunosuppressed and you come into contact with it, it can cause severe illness. It can cause some respiratory illness, and it can also cause meningitis. With AIDS patients, back when the epidemic was going on, we saw this very frequently with AIDS patients. This form of meningitis caused by this yeast. You'll notice here the colonies, can you tell they're a lot larger, and they're really wet and shiny, aren't they? We call that a mucoid colony. They're so mucoid because they produce this capsule. Just like Dr. Daft has talked about, bacteria can often produce a capsule that helps the bacteria. It gives it protection. It helps it adhere to cells, so it's both used for protection and virulence. And the same thing with Cryptococcus neoformans. It has this really large capsule. And we have a test we can do in the lab called an India ink prep, where we put a little bit of the yeast in saline on a slide and drop in a drop of India ink, which is a really dark dye. And the capsule is polysaccharide, and so it won't allow the India ink to penetrate there. So here is your yeast cell in the very center. All of this white area around that is the capsule. It's very large, so it looks like it has a halo effect in this India ink. I think we should do this in lab, Dr. Daft. Okay, so that's just something pretty distinctive about Cryptococcus neoformans. We can go on and do other enzymatic and biochemical tests on it as well, but that's something that we do if a patient does have meningitis and they have a lot of this yeast in their spinal fluid. We can take the spinal fluid, maybe spin it down and concentrate it, centrifuge it, and then do this test to see if that yeast is present in the spinal fluid. Okay, let's talk about the molds. So they are multicellular, where the yeasts are unicellular, and their main structure, kind of the body of their structure, is called hyphae. And it will increase in length. We say that it grows apically using mitosis, and it tends to grow towards a food source. It's when it tends to grow. The higher fungi, and which is really, I would say, I don't know, the majority of the fungi have these septations or cross walls in the hyphae. They are considered, I guess you could say, more evolved if they have the septations. It's an advantage for the fungi because if part of the strand of hyphae is damaged, on the other side of that septation, the fungi can remain intact and alive, and it will only kill the part on one side of the cross wall or septa. So there's a picture of hyphae, and you see there's a septation where they've drawn there. And you see how it can grow apically. A whole mass of hyphae we call mycelium or mycelia. Now, in addition to the hyphae that form, we also have some other reproduction structures, but let me talk about how the fungi uses its hyphae here to get nutrients. So if we're outside and got the soil here, let's say this is the ground, we've got a lot of soil right here, it's going to put down often almost roots, you might say. And this part of the hyphae is called vegetative hyphae. This is the part that is involved in finding nutrients from the soil. And this part up here we can call aerial hyphae. And this is the part that's going to contain the reproductive structures. Also on this part, it's not labeled, but you can sometimes have structures we call conidiophores that attach to the hyphae and that then give rise to the conidia. And I don't know if I said this, conidia is just a term for those asexual spores. So spores produced by the asexual reproduction process. They're a type of spore. And if also, well, you know, normally the fungi are out in the soil. If we grow them in the lab, this surface here, instead of being soil, that would be agar. So it would put down some of the hyphae, try to get it below the agar surface to absorb the nutrients in the agar. And then it would, what we would see on top of the agar, okay, sorry, let me go back. We would see this part of the hyphae, and with conidiophores, if they're present, and spores or conidia at the very top. And depending on how tall the hyphae is, it kind of, that affects if the fungus looks very cottony or fuzzy to us, or if it's more woolly looking with a shorter aerial hyphae. So for molds in the lab, this is kind of our procedure for identifying them. We read the cultures once a week. That's because they grow pretty slowly. And I had mentioned this with the yeast. They also grow a little bit slower than bacteria. They grow, it takes them about 48 to 72 hours on blood agar or fungal media. Molds take at least a week, sometimes 2 to 3 weeks to grow. Now, why do you think yeast and molds might grow more slowly than bacteria, based on what you've learned about bacteria and prokaryotes and their growth curve?
Oh, I was saying, like, because they produce by like mitosis instead of like,
Yeah.
and if they're eukaryotic, then like they have a nucleus and they're gonna have like, it's like the, I don't know, yeah. the translation transcription, like.
Right, right. So all those things that Dr. Daft has been over about the differences in prokaryotic and eukaryotic replication and transcription and translation, and how much faster prokaryotes are at those processes. So these are eukaryotic organisms, so they are not going to grow as quickly. They're going to take longer. The rate of growth, okay, can vary, even though they all take longer than bacteria. There is some variation, and that can give us a clue as to which mold we're working with. They can grow on antibiotic media, some, not all do. So media with antibiotics added into it, some are able to grow better. And then again, we looked at that colony morphology that I just talked about. The biggest part of identifying a mold is probably the microscopic morphology, because we're looking at those reproductive structures to help us identify those, and they can be really, really varied. A lot of variation, so it's pretty challenging to determine which molds you have. What you don't see on this list, you'll notice, is biochemical testing. There is some biochemical testing available for molds. There is not a lot. And I think one reason they haven't developed a lot of biochemical tests in the lab is because of their growth rate, probably. It just doesn't make sense to inoculate a mold to biochemicals because they grow so slowly. And it would be difficult to determine their whether they're positive or negative that way. So we haven't been real successful with biochemical testing with molds. We do now, it doesn't say it here, but we also do, not in most hospital labs, but in reference labs, they are doing molecular testing on molds for identification. The source of the specimen can be important because of the type of infection that you might have. So if it's from a respiratory source, you think about molds that can cause respiratory infections. If it's from a skin source, you think about that. We'll talk about the infections they cause in just a minute. Oh, in fact, now. Okay, so mycoses is a term that just means a fungal infection. And we divide them up into these 4 main groups. I bet you know what cutaneous means. Just an mycosis that infects the skin normally. Subcutaneous, it can involve more the dermis and the connective tissue below that. Opportunistic, systemic infections. So systemic would indicate that it has gone to multiple organ systems. Usually that means it is spreading through the bloodstream and can affect multiple organs. That term opportunistic means it's one of those molds that normally wouldn't cause us a problem, but due to someone having a compromised immune system, it may do that. And then dimorphic systemic mycoses. What do you think that term dimorphic means? Dimorph. What do you think?
Forms.
Sorry?
It has 2 forms.
Yes, very good. 2 forms. So you know how I've just told you that fungi are usually either unicellular yeasts or multicellular molds. We've got an exception to that. These fungi grow as a mold at room temperature and as a yeast at body temperature. So we'll talk more about that in a minute. They are more pathogenic, or in other words, they cause disease more easily than most of these other fungi we're talking about today. So cutaneous mycoses, normally just infections of the skin, hair, or nails. So they don't invade usually further down into the dermis or below that. Can you think of a fungal infection that is pretty common that we see on our skin?
Athlete's foot.
Yeah, very good. So any type of ringworm. So it could be the skin, the smooth skin, the hair, the nails, the scalp. There are several of those. So the proper term for that is dermatophytosis. And those fungi that cause those types of cutaneous mycoses are dermatophytes, and those genuses that are listed there: Microsporum, Trichophyton, and Epidermophyton. Candidiasis is also considered, can be a cutaneous mycoses. That's not very common, and usually, again, that's in someone that's immunosuppressed. But if you think about, especially the mucous membrane part, like thrush, sometimes too, another risk factor, I don't think I mentioned this earlier, for fungal infections, if you're on antibiotics, because yeast are part of our normal microbiome. And if you are taking an antibiotic, what is the antibiotic going to kill? What do antibiotics kill?
Bacteria.
Yeah, bacteria. That's also part of our normal microbiome. So once they're depleted, that gives the yeast a chance to kind of flourish, because they're not going to be affected by the antibiotic. That's why a lot of times you can get a thrush, or women can get a vaginal yeast infection from the yeast taking over after an antibiotic, after that you've been on one for a while. This is a picture of one of the fungi that cause ringworm, the Epidermophyton. This is how it looks under the microscope. If we grow it on an agar plate and do a prep on a slide and put that under the microscope, this is what we'll see. And you can clearly see the hyphae here, can't you? And you can even see the septations in the hyphae right there. This particular fungi forms conidia right off the hyphae. So there's no conidiophore structure. That's what that fancy term allurioconidia means, that it forms conidia directly on the hyphae. And you know how I've been talking about how fungi really, they're happy out in natural environment, and they don't really need us much. Well, here's one exception. This fungi is anthropophilic, which means that it loves man or humans, and it does prefer a human host. And there are several of the species of Trichophyton that are also really in need of a human host. So one of the few exceptions to that, these are some of the only fungi that really seek us out. Okay, so subcutaneous mycoses. These often start as the skin or a wound infection, but they will not stay just on the skin. They will be able to cause a pathogenic response deeper into the dermis and the connective tissue. So they're normally soil saprophytes. Like we've talked about, there are a lot of fungi that fit that category, but they are able to adapt to the tissue environment and elicit disease. So Sporothrix, this is a weird name, that Sporothrix shankii is the name of this fungus that is one of these causes of subcutaneous mycoses. It is often in the soil. They call this rose gardener's disease sometimes, because if you like to garden and you get really punctured with a rose thorn, or really it could be anything, but you get a puncture wound, allows those spores to get down under the skin, and they begin to elicit this type of response. And you'll notice that maybe, I can't be sure where this started, but it may have started here, and it tends to travel up the same pathway as the lymphatics. So it's going up the lymphatics. So our lymphocytes are involved in responding to this infection. And it elicits a strong response from our adaptive immune system. Here is how this fungus looks under the microscope. Isn't it pretty? It's hard to believe it would cause all that trouble. So there's your hyphae. This one does have a conidiophore, and then there are the conidia that form at the tip of the conidiophore. You notice some of them are able to form along the hyphae as well. Sporothrix tends to be more of a cause of subcutaneous mycoses, because it's actually dimorphic, which is kind of confusing. But it will grow as a yeast at body temperature and as a mold at room temperature. So it's able to be in the body. It can adapt to our body temperature very easily and cause infection. It doesn't tend to go systemic though very often, but it can in someone that's immunocompromised. So you could have a systemic infection with this one rather than just a subcutaneous. That looks worse than the Sporothrix, doesn't it? But this is another type of subcutaneous mycoses. We see this organism more in the tropics and the subtropics. I don't know if any of you have been on a medical mission to Guatemala or Honduras. I mean, it's possible that you could have seen something like this in a patient there. This is a patient who, I mean, clearly this infection has been going on for a long time. So, but we know in those sorts of countries that we find in the tropics and subtropics, a lot of them the patients don't have access to good health care, and they may let things go, because they just can't, they don't get to a doctor very often, if at all. And so this infection has obviously really long time, and the lymphatics have been involved here trying to respond to this and have probably been blocked by this organism as it grows inside the subcutaneous tissue. That's why you've got so much swelling right here. I think they call that elephantitis.
Professor Perry, does that have to be like treated and then surgically removed?
By the time it got to that point, I would say probably they would need some surgery, I would think. Yeah, if they let it go that long. This is the organism that was causing that infection you just looked at. Again, isn't it really pretty under the microscope? I think of this one kind of like a vase of flowers, because that conidiophore is shaped so neat, just like a vase. We don't see this organism as much in this country. It is more in the subtropical area. All right, so again, and I've kind of talked about this already. So mini molds are not very virulent, but they can still cause infection in patients with impaired immune systems. And these are some of the organisms that do that. We already talked a lot about Candida and Cryptococcus, the yeast. They are both in this category of causing disease in immunosuppressed patients. Aspergillus, a fungus mold that we see a lot. I mean, it's everywhere. It's very ubiquitous in our environment. And so it can cause a lot of respiratory illness. The zygomycetes, I'm not going to talk about much today, but they are very common in our environment. They grow on, we think of them as bread molds sometimes. A lot of times when your bread gets moldy, it's with one of those organisms. But they can also cause serious illness in immunosuppressed people. And those terms there at the bottom, that high-low hypomycosis just means molds that are hyaline or that lack pigment. Some molds have a lot of melanin pigment, and we call those dematiaceous molds. And so that last term, phaeohypomycosis, PHAE means dark, that prefix. So those are molds that have a lot of melanin in their structures, and they're very dark. They look dark growing on our augurs and also under the microscope. So a lot of terms there. Mycology kind of has its own language. I threw this slide in. This is actually one I took in the lab, so that's why it's kind of blurry. Because this is one of my favorite Aspergillus species, because you have, I don't know, it's a little hard to tell, but you can see these strands of hyphae, I'm hoping, running along through here. And these structures that are light brown, those are the conidiaphore that's attached to the hyphae. And then you have, this one's kind of upside down. Here is where the conidia are forming. And the conidia are the little greenish circles. Those are the conidia. Okay, you can kind of see them right here. So that's your asexual reproduction structure. And that's how we identify our Aspergillus. There's a bit more to it, but that's the general way. Now, this structure here, this is a sexual reproduction structure. Remember how I said we don't see those much in the lab, but this is an exception to that. This particular species of Aspergillus, for some reason, tends to form the sexual reproduction structures more. I think it may be the mating strands for it are just more prevalent in nature, and maybe. And so anyway, we do see those sexual reproduction structures more in this species of Aspergillus. This is called a Cleistothecium, if you are dying to know what those are called. It's a type of ascocarp, which is a sexual reproduction structure. Inside it are spores too. Just like you have the asexual spores here, these are the sexual spores inside this structure here. So I thought it was neat to see both in the same field of view. These little structures here, you see those really white, shiny looking cells? Those are called Hewley cells. We don't really know their function, but we tend to see them whenever we see this Cleistothecium structure. So we think they may have a function in forming that structure. Okay, the dimorphics. So remember I said they have a filamentous or a mold form that grows at room temperature, which is what's going to be out in the environment, isn't it? And then the yeast form is at 37 degrees Celsius or body temperature. So if you think about it, again, if we're out where these fungi grow in the woods or out in nature somewhere, and we come into contact with these spores, we're likely to breathe those in. So the infection normally, if we're going to have an infection from one of these, it's going to start in the lungs. These fungi, there's only a few dimorphic fungi, thank goodness, and they are geographically restricted. So they are only endemic to certain areas of the world and not others. Here's the list of dimorphic fungi. I'm going to talk a little bit about histoplasma today because it's the one that we have the most contact with in our area. But there are several other ones here that you see. Blastomyces is also found in the southeast. Coccidioides is found out in the southwest, and California, Arizona, Texas, and also in Central and South America. Paracoccidioides tends to be in Central and South America. Of course, I've already talked about Sporthrix. It is in our part of the country, and really I think it's probably all over the US. I'm not sure where it doesn't grow well, but it grows pretty much everywhere here. Penicillium marneffii is one species of penicillin that has the two forms of growth, and it is found in Southeast Asia. So again, with histo, there we are right in the hot zone for histo. It tends to grow well in this climate, in this moist, warm climate. It's very happy in the Ohio and Mississippi River valleys. So if we were to grow histo in the lab on some agar that it likes and do a prep and put it under the microscope, this is what we would see. So you see the hyphae here, all the strands, and then you've got two types of conidia, don't you? These form right on the hyphae. There's no conidiophore, and we call these just microconidia and macroconidia. And another really distinctive thing about histo, do you see these little kind of spikes around the macroconidia? Kind of reminds you of coronavirus, doesn't it? These are, we call those tuberculated macroconidia because of those growths on the outside of the conidia. The yeast form that grows in your body just looks like a little yeast, doesn't it? Not too descriptive. It's about the size of Candida. Now this is another mold that looks very, very similar. In fact, I couldn't tell this one apart from a histoplasma. It's called Cepidomium, and it is not very pathogenic. So in order to distinguish histoplasma from Cepidomium, we have to either convert it to the yeast phase to prove that it's histoplasma. We don't normally do that anymore in the lab because it is very time consuming and tedious to try and get it to convert to the yeast phase. You have to put it on a special media and incubate it at 37. Usually takes several tries. So we do molecular testing now to identify it definitively. And that GenProbe test is a really good test. It says false positives can occur, but it's pretty, that's pretty rare. So like Cryptococcus that I talked about earlier, histoplasma also likes to grow in soil with bat or bird droppings. That gives it, I guess, a lot of rich nutrients. And then it'll circulate in the air when the soil is disturbed. And then those spores are inhaled into the lungs. And because of our body temperature, the spores convert to the yeast phase. And that is then it begins to cause disease. Normally, it's going to be encapsulated by macrophages in our lungs, but they may not be able to contain the infection in certain cases, and it can then spread to lymph nodes and other organisms through the bloodstream. These are the symptoms. If a person becomes really ill with histoplasma, you would see these symptoms. They're kind of vague symptoms, but do they remind you of any other disease that you may have studied about in the past? Especially the cough, the night sweats, the weight loss. Does that sound familiar at all to anything else? What about TB? Does that sound like a person that would have TB might have those symptoms? They would. They would have almost identical symptoms. And so the fact that the symptoms are the same, and it can also be when the lungs are looked at, the lungs can look similar due to the pathology of the organisms being phagocytized by the macrophages. So doctors sometimes are confused about which illness a person has. Because of the pathology and the symptoms, they can look very similar. So this is just telling you that most people that come into contact with histo are going to be fine. They're not going to have much of an illness. And in fact, because we all live in this area, if we were to go and have our lungs x-rayed right now, most of us, they might be able to see a little area in your lungs where you've got a little scar from having histoplasma, even though you were never even aware of it. Most of us have that. So it's only with patients that are immunosuppressed or that really get a really large concentration of spores, if you were to breathe those in, that you would develop this chronic disease. People that like to go, is it spelunking? Is that the word? That like to explore caves. If you think about somebody that's down on the floor of a cave, crawling through the cave, and remember bats are in the cave, and their droppings are in the cave, and the soil in there, that's a great place for histospores to be, and they might be in a fairly high concentration. So if you're down on a cave floor crawling through there and disturbing that soil, you could really get a high concentration of those spores and develop histoplasma. I knew someone in college that did that, and he was otherwise healthy, but I think he got such a high concentration of the spores that he did develop histoplasmosis and was sick for several months. If it does become systemic, those are the organs that it tends to spread to. And there is a lovely picture of someone that has had histoplasmosis spread through their bloodstream, and they have the lesions inside their mouth. Okay, so as I was saying, macrophages, that your body's immune response to histoplasma is that macrophages are going to phagocytize those cells and try to contain them if they can. Sometimes they will form giant cells to try and contain them. So they'll have several macrophages fuse to form giant cells. And also T cells will come to help. We haven't talked about that yet, but I know you guys have studied immune system before. So when those T cells and macrophages kind of fuse, that forms a granuloma. And again, it's just a way our body tries to contain the yeast cells that are invading. That term parenchymal necrosis, that just means that the tissue in our lungs can be damaged and dying, and we're going to have a loss of function due to this infection sometimes. And this is just a picture showing lung tissue, and here, this is probably a granuloma forming. The yeast cells there are really clearly visible. And this is just another stain where the cells are blue rather than dark, but same sort of thing going on there. So I tell this to my nursing students. You know, nurses especially have such a big role in patient education. So patients who are immunosuppressed really should avoid activities where they are going to come into contact with high concentrations of histo, because it is pretty pathogenic. So those are some good things to avoid if you are immunosuppressed. All right, I think I've gone over, Dr. Daft.
I mean, do you have any more? Do you want to go a little bit longer?
Well, I could talk about coccidioides if you would like, but that's totally up to you.
Well, how about, would you be okay if we take like a 10-minute break and then I'll let you finish up?
Yeah, if you want me to. Now, I don't have to talk about cocci unless you just would like me to.
I think I find it pretty interesting. So I'd say let's take a 10-minute break-ish. So 9:08, let's come back at 9:20 and we can finish up, and then I'll just use whatever time I have left. All right?
Okay, sounds good.
All right, guys. 10 minutes-ish, a little more.
The screen again.
All right.
Okay, well, so we'll just talk about one more of those dimorphic fungi. This is probably the most pathogenic, I would say, of all the fungi that we know about. But it's similar in its pathogenicity to histoplasma. I would say maybe just a little bit more pathogenic than histo. This one, thankfully, is not in our area of the United States. As I was saying, this one tends to be in the Southwest. It likes the really dry, hot conditions. And so also, of course, down on into Central and South America, we find this fungi, and in other parts of the world too, but around in this part of the world, this is where it lives. And so it likes the dry soil and dust. And so when those spores are disturbed and inhaled, you can get this illness. They call it valley fever. So again, it's the same sort of process. The spores reach the lungs, and because of our body temperature, this time it's not really a yeast that forms at body temperature. It's a spirale, and I'll show you a picture in a moment, and with spores inside the spirale, the endospores. And then those endospores are what's infectious in our body and that can potentially travel if they're not contained in the lungs to other parts of the lungs or through the bloodstream to organs and form more of those spirals. I haven't shown you any pictures of what molds look like growing on the agar, so this is showing you how it looks growing on fungal media. It is normally a white mold. It is very cottony texture, so it has a fairly high aerial, the aerial hyphae is fairly high up off the agar plate. What we call the reverse, if you flip the plate over and look at the underside of the colony, it tends to have a little bit of pigmentation. It tends to get, especially with age, it will get a little bit brown. So this is a picture that I took in my lab in the last hospital I worked in Nashville. You know, again, I said this is endemic to the Southwest, so we don't expect to see it very often here, but people travel. So this was a patient who was actually from Chile, I believe, and I had moved here to Tennessee, and I was certainly not expecting to find this when I did my tape prep. Because one of these little spores here, we call these arthrospores, is enough to cause disease, just breathing one of those in. So in the lab, this is something that we try to be very careful with. We always, and I probably should have mentioned this earlier, when we do tape preps, work with a mold that we've grown in culture, you are always under the biological safety cabinet to do that, to prevent the spores from coming out into the air where you might breathe them. And sometimes too, we wear an N95 mask. When we're taping something like this that's highly pathogenic, we want to be as careful as possible. So these, like I said, these are arthroconidia, and you have a spore, and you notice these lighter areas. So those are kind of just a connecting cell. Those aren't the actual spore. They're just we call them disjunctor cells. It's very distinctive looking. There aren't a lot of fungi that have these arthroconidia, and these tend to be somewhat swollen too. So that helps us think about coccidioides. So that is the mold phase. And then in the tissue in our bodies, these spherules will develop. And you'll notice these two over here on the right are just starting to develop. And then we have one over here on the left that's really mature. And those endospores, as you see, they are kind of being released. There, the spherule is breaking open and releasing the spherules. I'm sorry, the endospores from the spherule. So remember with histoplasmosis, I said that 95% of people exposed to histo were probably just going to have a benign, minor infection. But look at the statistics here. 60% have the benign. 40% develop more serious symptoms. So that's why I say that this one is a bit more pathogenic, able to cause disease a little easier than histoplasmosis. And so they begin, if they do develop the serious symptoms, it begins with a flu-like illness. And then some people will go on to have dissemination to other parts of the body, the meninges, the bones, the joints, subcutaneous and cutaneous tissues, even the skin. Within the first few weeks to months, it varies a good bit how your body responds, I suppose, in fighting the infection. Obviously, people who are immunosuppressed are going to be much more at risk with this fungus. let's see, there's where it has spread to the skin. So that's just, I don't know, that's pretty amazing to me, the way these dimorphic fungi cause disease. If you saw this patient, that would probably not be the first thing you would think of, was that there was a fungal infection that had started in their lungs. But that's what's happened here. So there is just some statistics on the incidence of coccidioides. I'm not sure what happened there in 2011. Maybe we had some sort of weather-related thing that caused more people to be exposed. But there is fairly high incidence of infection there. That I don't know, I guess you can see. So the blue is Arizona, and then this orange is California, and then the purple is all other states where it's reportable.
Professor Perry, this is kind of random, but I was curious, do you know anything about age demographics of cases, and is there any seasonal relationship? And I just mentioned that because I'm curious, because I know, Arizona, where this is really prevalent, has a lot of older folks that go there winter. And so do you have any idea if it's more prevalent in older folks?
Well, I don't have the data on that. I bet there is some. I could look for it. I would say, because older people, their immune system obviously doesn't respond as strongly. So I would say, it's possible that it is more prevalent. That would be probably one of the higher risk groups, I would think. Yes. And as far as the seasonal, it likes hot, but Arizona, isn't it hot all year round?
So by seasonal, I meant, I realize temperature is the same, but if you have the snowbirds that all rush there, during the winter.
Yeah.
And so, there's a higher density of older folks, I would say, there from winter to spring before they head back. And so I was just curious, is there a spike in the winter and the spring?
Yeah, that would be interesting to look at. Yeah, I'm not sure. The dust storms out there, I don't know if they occur more in the summer. I really don't know much about the Arizona weather, but I think dust storms are really a good way to get exposed. I got to go and listen to a doctor. She is the director of microbiology, I believe, at Cornell University, and she published one of the books we use a lot in mycology. Her name is Devise Lerone, and I got to hear her speak, and she talked about going to visit a cousin in Arizona, and they went out shopping, and there was a dust storm. And she said, the whole time we were out, I was telling my cousin, I'm going to get coccidioides. I'm going to get coccidioides. And her, cousin probably was thinking she's a weird science person. But so she went back to New York, and in their lab, just like a couple of weeks later, maybe just a week later, they had some sort of exposure or potential exposure to coccidioides, where they were working with the fungus, and it was a potential exposure. So they tested everyone in the micro lab, and so they were going to get a baseline. They went ahead right then to get a baseline test, and then they were going to go back and test them again about 6 weeks later to see if they had been exposed. And her baseline was positive. So maybe it was longer than a week later that she was out there. But anyway, she felt like she had been exposed during the dust storm. But luckily, she didn't get any symptoms. There's some other statistics on valley fever. So it really does impact the healthcare system and patients. that in metropolitan areas of Arizona, 15 to 30% of community-acquired pneumonias are caused by valley fever. That's pretty significant. The deaths aren't terribly high, I guess, but does cause a lot of illness. Okay, and so there's just some things to help people not contract this. I had another case when I was at St. Thomas where we saw those spherules in the lung tissue. I mean, they were just textbook. They looked just like those pictures I showed you. And he was from Mexico, the patient was. He did have AIDS, and he also was a construction worker. So it was kind of a perfect storm for coccidioides. All right, I think that's it.
Recording. All right, we should be good here. All right. So, all right, we just got to horizontal gene transfer. And again, I'm hoping you spent some time reading through the paper, because if you did, then a lot of this makes sense. And this will simply be review, right? And that'll be good because hopefully it will stick there a little bit. So we're talking about horizontal gene transfer, right? We're referring to cell-to-cell movement, all right? And for a horizontal gene transfer to officially be considered legitimate or be considered to having actually occurred, right, that daughter cell or that recipient cell must hold on to the DNA that it receives. What we're going to focus on primarily is homologous recombination. And so if you remember previously, we had discussed briefly non-homologous when we talked about those transposons. And so basically, if you have a, we use the example, if you have a gene for hair color, it gets inserted in some random place that's nowhere near another gene for hair color, right? But what we're going to look at is homologous recombination, right? So exchange between two similar regions of DNA. And this, again, is a relatively highly conserved process. Now, one of the things, and sometimes I think I do a poor job doing this, is that when you receive this new genetic information during horizontal gene transfer, you're not keeping the recipient information plus the information that came over. You are replacing that segment of DNA in the host, or the host cell. In this case, we're talking about bacteria, right? And so the product that it receives replaces, right, a somewhat similar sequence that was already there. Okay. So again, very, very brief summary table, even more brief, I think, than what's in the paper. But remember, transformation, we'll talk about each of these in just a second, transduction, and conjugation. All right. Those are the three modes of horizontal gene transfer that I'm most interested in. There was another one in your paper that was very similar to transduction. So let's talk about transformation. And you guys answered this pretty well when we were discussing the paper, right? This idea of naked DNA. Its DNA can either be secreted or is likely the result of a cell that has lysed, okay? Maybe it has been killed by a medication. Maybe it's even been killed by a bacteriophage, right? There's a lot of things that could lead to that cell lysing. So you see here, right, this cell has to be competent, right? And bacterial cells can express receptors that allow them to grab onto DNA in order to pull it into their cytoplasm. Notice here when you look at the picture, it's only taking in one strand of that DNA, right? And so when that one strand enters, it then finds the homologous area on the bacterial chromosome, right? The original section of DNA on that chromosome, right, is excised, taken out, and we insert, right, the new segment of DNA. And now in subsequent divisions, right, you're going to have the presence of that new segment of DNA. And so here it was using the example of a streptomycin resistant gene that was transferred to these bacteria, all right? Yeah. Thank you. You're the best. All right. All right, bye. So that's my wife dropping off my hot tea that I forgot. So in this case, right, that information it took up allows it to be resistant to bacteria. So here you see two cells, one that took it up, one that didn't, right? So this was a division of the original cell before it took up the gene. You treat it with antibiotics, it dies, and those that picked up the resistant gene are now resistant to that antibiotic, right? Again, when we talk about this idea of competence, tightly controlled, right? Population density, nutrition availability. So this idea of there has to be some type of stressor, right? That could be, right, like we said here, some type of nutrients. It could be, though, the introduction of an antibiotic. And when that stress is present, right, those cells, if they're capable, right, become competent or are willing to take up DNA in hopes of finding something that may be helpful in their survival in that changing environment. Here is one of my, I think, probably my favorite type of horizontal gene transfer, and that's transduction. I think this is really cool because it's using a bacteriophage or a virus as the vector for this process. And so here you have a bacteriophage. Right? What they're not showing for some reason is, right, they do have those little kind of spikes that looks like the legs on a lunar lander, if you've looked that up, right? It attaches to the bacteria, inserts its genetic information. So the green is viral DNA, the purple is the bacterial DNA. When that viral DNA is inserted, it results in the lysine of the bacterial chromosome, right? And so now you have a broken up bacterial chromosome and you have the viral nucleic acid here. The viral nucleic acid takes advantage of the cell machinery. And so all of the enzymes that are used to make copies and to make protein and to make RNA and all that fun stuff are used by the nucleic acid of the virus. And so you start to see copies made of the virus. You start to assemble. This part of the virus is actually called a caspid. We'll talk about that next week. So the caspid is kind of this external structure of the virus. And then you package the nucleic acid in there, and the cell lysine bursts, and the bacteriophages go on and infect other bacteria. But here you can see, in some cases, you have this little thing down here called the transducing particle. And this is actually bacterial DNA that is accidentally packaged inside of a caspid, of a viral caspid, all right? So when it bursts, right, it acts just like a virus. It's attracted to bacteria, except if you come up here, right, now when it attaches and inserts, the bacterial DNA enters another bacteria, and instead of lysine, the whole chromosome, right, it excises the piece of homologous DNA, right, similar genes or similar areas on the chromosome, all right? And now, all right, as the bacteria divides, you see that the daughter cells, the subsequent daughter cells, now have that same piece of genetic information inserted into them, all right? We'll talk about this, this more a little bit. This is something called a lysogenic life cycle, all right? We'll talk about something called a lytic life cycle also that can do some interesting things to the bacteria, but that'll be next chapter. So again, presumably, right, maybe this bacteria was resistant to antibiotics. It gets a viral infection though, right? It gets broken up and lysed. Maybe that gene that has antibiotic resistance accidentally gets packaged in this capsid of this virus, and then passes that information on to a recipient, and you've now, right, created potentially more viral, or I'm sorry, more antibiotic resistant bacteria. And one of the things that's really interesting that we're starting to appreciate about bacteria is that horizontal gene transfer and the sharing of this genetic information can occur amongst different species of bacteria. All right, so it's not just like bacillus to bacillus or E. coli to E. coli or something like that, right? It's possible that you have two very different bacteria, right, that may be able to share genetic information, which is kind of fascinating from a scientific standpoint, but is scary, I think, from a medical standpoint. All right, so here, right, you see conjugation taking place. So here you have an F pilus or a fertility pilus, right, a modified fimbriae, right? So here you have the donor cell, and here you have the recipient, right? And so sometimes we unofficially refer to this as bacterial sex, right? The direct passing on of genetic information. Now, again, please note this is very different than sexual reproduction that occurs in fungi, right, that Professor Gray was just talking about, or occurs in our cells, right, when you have a pairing of two sets of chromosomes. Again, here for the most part, this isn't an addition or a replacement, right? You're not pairing things together to get a functional organism like you see in true sexual reproduction. All right. So here it's just giving you, this is just kind of a diagram from your book showing you what's going on. And so you have the donor cell, you see F+, so think of that fertility pilus. This is kind of important to note because you see this in F plasmid. So not all plasmids can necessarily be transferred over via conjugation, right? It's also, we'll talk about chromosomes here in just a second, right? But again, it's specialized, and that's one of the things that I hope you're appreciating. You need an F plasmid, right, to send whatever's on this plasmid via the F pilus to the recipient or F negative cell, all right? And what's really cool, right, is as you send it over, right, it's being replicated as it moves over to the recipient cell, right? So the donor cell holds on to, right, it's the original piece of genetic information, right? You make a small cut and you start to transfer over, and as it enters the recipient cell, right, you start to add the other nucleotides on it to give its double strand, right, or those anti-parallel strands. At the end of the transfer, right, you now have two F positive, right, cells that are capable of utilizing a fertility pilus to transfer genetic information, right? Now, I have it here. Yes. So what would be your guess, right? I show the plasmid here because this is the most common. Why do you think chromosomes aren't commonly transferred during conjugation? What would be your guess? Don't overthink it, right?
Would it be because they're like so big and like the pilus could be like so weak that it could break in the middle of it?
Right. That's perfect. You know, a lot of times, and right or wrong, maybe more wrong, we kind of approach this from a laboratory setting where you think something's growing in a culture or it's growing on a plate, right? And those are pretty ideal environments for a lot of these organisms. And, you know, truth be told, if this is occurring, especially out in the environment, outside of a petri dish or outside of a test tube, these cells might not be joined together for a long time, just like Rachel mentioned, all right? And so to get a whole chromosome over, right? So you think about it, and you remember how big, I showed you that image of that E. coli that we treated with some chemicals to make it cough up its chromosome. It's really big. It takes a lot of time to transfer that over. And typically, right, those cells are not going to be joined long enough for a complete chromosomal transfer. Now, I think if you look through your textbook, you'll see some images. There's a bunch of different possibilities. Sometimes you might get part of a chromosome over, and if you do get part of a chromosome over, it might actually insert itself into a homologous region of the recipient chromosome. But plasmids, much smaller, a lot less time to transfer them over, so it's going to be a lot more common for that to occur, right? So in this case, right, the size, right, of that genetic material really does matter because bacteria do not have a lot of time to be stuck together in order to exchange this information. Okay. Good. This is just a really brief slide that talks about plasmids. When we talk about plasmids in bacterial cells, first of all, remember they're accessory, so they're not necessarily present in all bacteria. All bacteria don't need them, but it is relatively common to have at least one plasmid in a bacteria, and sometimes they can have up to 500 plasmids present in them, right? And so there can be a lot of genetic information that exists outside of that chromosome that could be present in that bacteria. Host range specific to broad. So this is what I'm saying is that when we're sharing, some plasmids, are going to be commonly shared between two bacteria of the same species. But as I just mentioned, via conjugation, you can have a pretty broad range as to what can accept a plasmid from a donor, right? So you could be donating to a totally different species, totally different genre, or totally different genus, all right? And again, this is just showing, right, one of the common plasmids that we worried about is the R plasmid, right? That R stands for resistance. And so if we're talking about microbial resistance, right, that information is going to be found on R plasmids. So that's just a special designation that we give to those plasmids. So this, for me, is not something that I necessarily, I'm not going to test you on this. There's not going to be a genomic island question. But what's really cool about this, right, and if you know anything about genetically modified organisms, right, this is something that already exists. And this idea of taking large segments of something's genome and inserting it into a completely different species. All right. So here you have a Bacillus thuringiensis, and this is an anti-pesticide, or a pesticide, not an anti-pesticide, a pesticide, a naturally occurring pesticide produced by this bacteria. So you insert that part of the genome into corn, and then you have corn that is naturally protected, right? And again, I'm not here to argue GMOs. I'm just saying this is kind of where we're at. And I keep on saying this, and at some point I'll have Professor Perry hold me to it. One of the cooler examples that I've heard of, and I need to go and look it up, is that some groups have actually experimented with inserting small segments of proteins into fruits like bananas. And so you could actually vaccinate by eating a banana, right? And so taking that component of a pathogen that you would use in a vaccine and actually, right, if it's a certain protein, inserting that into the genome of some fruit, you eat the fruit, and then you get naturally vaccinated that way. So there's some really cool tools and implications. This is one of my favorite as a former type 1 diabetes researcher, although and all the time that I did type 1 diabetes stuff, I did very little actually with insulin. But right, this idea that we use E. coli to produce human insulin. So we've taken the gene for human insulin, inserted it into E. coli. Bacteria grow very quickly compared to growing or keeping something like livestock, like cows, like animals. It's incredibly cheap, right? So this is a really efficient way for us to grow and harvest human insulin, all right? Yeah, I'm not really worried about these. We've already talked about some of these before, again, so you can look at those if you want. We really only have a few minutes. Let me talk about really briefly about algae and maybe a couple of protozoa. I promise it won't hopefully take too long if I can figure out. There we go. Shut that down. Yep, I stopped sharing. I realize that. Okay. All right, perfect. So, this is actually kind of a cool chart, because when it comes to microbial eukaryotes just in general, and Professor Perry actually hinted at this when she was talking about trying to classify fungi, and a lot of the classifications that we're still kind of using for microbial eukaryotes comes from visual observations, right? And Professor Perry mentioned, they have this sexual reproductive structure. They have the asexual reproductive structure. And for things like fungi in the past, sometimes things that were the exact same organism were classified as two different organisms because they saw two different structures. And for algae, one of the things that happens is it's not a plant. It doesn't look like it's an animal. And so then we just group it all together, right? And we do some of the same stuff for protozoa. And so here you can see this is a phylogenetic tree. And the thing that I kind of like about it is in this case, right, I highlighted in red, these are all different types of algae. And so what you can see from a genetic standpoint, a lot of these things are not that closely related. And so our classification system is definitely outdated. But then you run into the issue of people that have been using this classification system for a long time, getting them to switch over and getting the field to switch over. Is it really worth the fight? I don't know. I'll let you guys decide if you decide to go into the fungal, algae, protozoa classification system. Also note here, I think this is actually really cool. You see in the phylogenetic tree, here's a bunch of different fungi, and then what do you know? Kind of right next to it, not too far away on our branch, is animals. And so from a genetic standpoint, we're actually really closely related, to fungi. So I just think that's, some of that genetic stuff is pretty cool, and it's maybe above all of our pay grades, but that's fine, right? So algae, right? Simple photosynthetic eukaryotic cells. So if you've hung out at the lake, if you've hung out around ponds, for some reason, maybe you like bogs, I don't know, right? Even the ocean in some cases, right? We see algae. A lot of things that you guys actually classify or would call seaweed, if you've spent time in the ocean, is not actually an aquatic plant, right? It's actually algae. And sometimes this gets a little confusing because they can be unicellular or multicellular, right? They lack organized vasculature. Those of you that have taken Bio 2, algae are kind of the precursor to land plants, and there's specific species of algae that start to take on some of the characteristics that we associate with land plants. And so we kind of consider certain algae kind of that link species between aquatic vegetation and terrestrial vegetation, right? So like I said, the majority here are aquatic. I have it on my last slide. It's definitely not as cool as pathogens, but fungi can actually form some really cool, unique relationships with certain algae depending on the environment. So if you've heard of lichens, which kind of has this mossy-like appearance, sometimes that can be algae that's formed a symbiotic relationship with fungi. Photosynthetic, so that's a high energy source, and so that's an attractive companion for some fungi. And then fungi can bring in other nutrients, and so you get kind of cool symbiotic relationships between some of these microbial eukaryotic cells. All right, algae, one of the major components that they use to capture this radiant energy is chlorophyll A. They have cellulose in their cell walls, and a lot of them, right, because they're photosynthetic, are capable of carbon fixation, right? So you have multiple microbial cells, right? Prokaryotic cells and eukaryotic cells that can fix carbon. And here's just a couple different pictures of algae. And you can see here, right? It's fascinating. These are all algae, and they're all very unique, even morphologically. Again, I think I have a video here on a type of green algae that is actually toxic to aquatic life. And so we have some species of algae that are very invasive. These are the major groups of algae. Some of these, right, if you like seafood, if you've had seafood, or if you've had sushi, right, that's wrapped in seaweed, right, that's actually an algae. If you brushed your teeth, if you would be in microbiology lab and we were using agar, right, those are algae byproducts that we take advantage of. If you're buying ice cream, right, and it's been processed a lot of times to kind of make it a little thicker and hold its form, there will be algal components in it. Probably notice ice cream from a creamery is sometimes different than ice cream in a box. And that's one of the reasons. So again, I just kind of wanted to briefly go over those. Someday, maybe I'll pay more homage to algae. From a pathogenic standpoint, which I think is, one of the things that we're often interested in, is this is something called red tide. And so you can see that forming in the water here, right? This is formed by a type of algae called a dinoflagellite. Right, here you see another algal bloom here. And then here, what you can't see is that Professor Perry's swimming right over here. She just didn't want to be, caught in the, right? I don't know. This guy to me, there's been some water that I'm like, should I get in? Should I not get in? I don't know. I guess if you grow up around it, but this is not where I would be swimming. All right. This is a river, I believe, in China, right? And so you can see this is a really bad algal bloom. This actually to us is not directly toxic. Where we run into issues is if we eat any type of aquatic life that's been in that water, then the toxins can be passed on to us and can cause a number of issues. These toxins typically are going to interfere with signaling within muscles, and so there can be paralysis and other associated symptoms, right, which becomes a huge issue if you're talking about a muscle like your lungs or the diaphragm, right? But unless you're eating stuff from this water, not really that big of an issue. Now you see here this huge kind of population. You can see this is a big city. We briefly mentioned nutrients for bacteria, right? Things like phosphates, you could even argue things like iron, all that type of stuff. If a lot of that makes its way into a water source, then you can have algal blooms like this, right? And so this is not what I would call natural. In this case, right, something's probably polluting the water. That's a great nutrient source for the algae, and then you have an algal bloom. If you talk about aquatic life too, this can be an issue depending on the type of algae, right? It's not just dinoflagellates. But if you have a water source that has an algal bloom, right? Algae, consume nutrients from the water. They can deplete gases from the water that are needed by that aquatic life. And sometimes with algal blooms, you'll also have mass die-offs of aquatic life because there's a direct competition for the nutrients that are in that water. All right. So what I'm going to do is I'm going to end there because I don't want to keep on going into the protozoa. If you want to, and we may still discuss this next week, you can see that there's a few slides on protozoa. Again, just like the algae, you can see that they're kind of classified all over the place. They do a lot of cool different things. So if malaria, right, is caused by a protozoa. One of the diseases that you guys could choose, I'm just kind of clicking through here, right? Trypanosoma cruzi, right? So if you've heard of Chagas disease, this is a kissing bug. What this bug does is it bites you kind of on your cheek or face region. It then defecates near the bite mark, and you go to scratch that bite mark because it's itchy, and then you basically pop the protozoa into your bloodstream. And then this is the tsetse fly. It causes African sleeping sickness. There's two versions of this. And so you get bit by one of these flies and the protozoa enters your system. All right. So again, we may talk about these more next week. These are definitely some really cool, interesting diseases, especially these two, that there's a good amount of information on, if any of you are interested in using that as your microbial disease for your presentations. All right. So I'm going to go ahead and stop, all right, this share and this record.